Hi, Linda. When we talk about cardiovascular medicine, for me, this was really my introduction into the cell therapy space. I started looking at the work that Athersys was doing, that Neostem was doing, and then that Mesoblast was doing, and I was very excited because for me, it's not really a question of if it works, right? I want to say that again. It's just, for me, it's absolutely not a question on does this work. I know it works. You have to prove it clinically. But when I met Dr. Marban on a trip to Cedars-Sinai and I visited her lab and I saw the science and I saw the technology, she said something to me that I thought was, just made perfect sense. It was, how do you speak the language of the heart? How do you expect a mesenchymal cell to really be able to optimize the language of what's happening inside the cardiac muscle itself? And I started to think about what always happens in the world of drug development. When Merck introduced Mevacor, it was an amazing new drug, but it was the first of several drugs. And I remember when Pfizer, then Warner Lambert, introduced Lipitor, and I thought, can the market really expand? And the answer is, not only did Lipitor expand the market, but the market went from $2 billion to $10 billion. So, now we're not just talking about does cell therapy work in the heart, but we're actually talking about what I view as a second or even third generation cardiac-based cell therapy in Capricor. And there's lots of data and lots of things that I want to talk about with you in the next 7 minutes. But one of the things that also interested me was the fact that you've been able to attract Johnson & Johnson, and you've really been very successful at building this company. So introduce us to Capricor, and we'll talk a little bit about what it's all about. Thank you.
So obviously, Capricor is a publicly traded company, and this is our forward-looking statement in case I make any comments of that sort. Go. So Capricor is a really unique company in the regen med space because we're a diversified company that is providing therapeutics for the development, for the treatment of heart failure. Okay, thank you. And so we have 3 products in our pipeline. The first product, the one that we'll probably talk the most about today, is our cardiosphere-derived stem cell product called the CDC. That one is in a phase 2 clinical trial, our all-star clinical trial, and in the dynamic clinical trial, which I'll talk about in the next few moments. Our second product is a non-regen med product, but one we are equally excited about taking into the heart failure space. It's called Cenderitide. It's been in 270 patients, and we are just now wrapping up a phase 2 pharmacodynamic, pharmacokinetic study where we're partnered with Insulet to deliver this technology to patients that are coming out of the hospital after having a heart failure episode in order to stabilize them at home. And perhaps this therapeutic would be a nice bridge to using with the LCZ696 compound that's most recently been shown to have profound effects on mortality in patients that have early stage heart failure. And finally, our third product, which is still in the lab, although look for it to come out in a very pretty dress in the next year or so, is our microRNA delivery platform, our exosomes. Exosomes perhaps mediate the potency of our cells, and were discovered actually in looking for a potential mechanism of the action of the cells. Capricor has $25 million in the bank. We just did several fundraisers through a PIPE that has allowed us a 2-year runway. We have a license option on our cells from J&J, as Jason mentioned. We'll probably talk about that. And we have strong operational leverage where we've brought in almost $40 million of non-dilutive capital since our company was founded. Now let's talk about regen medicine because that's what we're here today to talk about. We have 3 trials, 2 of which are actually underway. The All-Star clinical trial program is treating patients that have cardiac dysfunction after a heart attack from 30 days to 1 year. We anticipate the data coming out from that trial late 16 or early 17. It's actively enrolling as we speak at 25 sites across the United States. The dynamic clinical trial will be wrapping up in just the next few weeks. We announced a few weeks ago that is a trial in which we are taking our cells to those patients that have advanced heart failure. These are people that have class 3 or ambulatory class 4. That means they're pretty darn sick with their heart failure. We're using a different delivery method. Our delivery method is intracoronary. We like to use a very easily accessible delivery platform so that cardiologists anywhere in the world can deliver this therapeutic. We're using triple vessel delivery in this situation where we're bathing the heart in the healing cells because at this point the whole heart has now been damaged. And then finally, and I think many of you have seen that we've announced that we're going to be treating the cardiomyopathy that's related to Duchenne Muscular Dystrophy with our cells. Our cells are uniquely suited to treat this type of disorder because it's a fibrotic disorder where the cells die and the heart necroses, and ultimately these boys and the parents will tell you they quote-unquote fall off a cliff because their hearts get so stiff and fibrotic. And so we had a meeting with the FDA just a few weeks ago, and we're preparing an IND, so you should see this trial initiated in 2015.
Okay, so before you go off the slide, I would like to get out a couple of questions. When we talk about a second-generation therapy, right, tell us a little bit about, is this autologous, is this allogeneic? Have you lived through that decision? Because I think that's very differentiating. Tell me a little bit about the mechanism of action by which you deliver these cells and how they might act differently, and then let's talk a little bit about the difference between MI and heart failure.
So thank you, Jason, for that. So we started in the world of cell therapy with an autologous product. The first trial that we did was called Caduceus, in which we treated patients with cells taken from biopsies from their heart. So our cells are actually made from cardiac muscle, and Jason alluded to that when he did his very kind introduction of me. He showed what we showed was that we were able to see a marked reduction in the amount of scar in the heart after the heart attack. So when the heart has an injury like a heart attack, there's a scar that's created. The Caduceus trial showed that we had a reduction in the amount of scar. However, we decided that an autologous program was just not one that would be very easy to develop from a commercial standpoint. So we decided to go back to the bench and try out an allogeneic type of cell. So we take our exact same manufacturing paradigm for the autologous, but now we make an allogeneic cell. We've tested that in humans and in animals. It's non-immunogenic. There are no clinical sequelae that are related to a potential immune response. And we've shown now, at least in a proof of concept way, that we can deliver these cells safely to humans, which was the phase 1 All-Star trial presented last year at AHA and at ACC.
So you made a decision to move away from autologous towards allogeneic because you believe the cells are not integrating, they're just exhibiting a trophic effect, and you didn't see any problem with immune rejection in that transition. Is that fair?
That's right. We like to think of ourselves as a local drug delivery system. So they go in and they release a lot of factors that stimulate endogenous regeneration. So we've shown, at least in preclinical models, that the cells from the heart actually replicate themselves. They divide and cause the scar size to be reduced and new tissue to be created.
And why use heart-derived cells? I mean, is there something different? Do they really have a different language? Do they have a different cytokine expression profile than, say, a mesenchymal cell, so that you can help me understand that decision?
So our cells are a completely unique cell type. We've done a marker panel where we look at 166 different extracellular markers, and they are completely different from any of the other mesenchymal or blood-derived or bone marrow-derived cells that are being used to treat any disorder. We believe these cells have unique healing potential, and we also believe that they're uniquely suited to the heart, although preclinically we see that they work other places as well, and we're not talking about that right now, but they're very powerful healers.
And what I see on this slide are two very, very different trials. The mode of delivery in these trials are both infarct-related artery, but I guess All-Star would be IRA. What would I call dynamic? It's not localized NOGA delivery, is it?
No, so speak often about the fact that we think that the best way to get a therapeutic into the heart is using the vascular delivery system provided by the coronary arteries. So all-star, we're going down the infarct-related artery, as Jason just said. In dynamic, we're using triple vessel delivery, and that's also the strategy that we're using for the Duchenne Muscular Dystrophy trial. So in dynamic, we are perfusing the heart, and it's been a dose escalation study where we want to see to tolerance how many cells we can put down the heart. We're in the last cohort of that trial right now, where we're putting 25 million cells down each of the relevant coronary arteries, and there's 3 of them. We'll be using the same strategy for DMD, and the nice thing is dynamic will allow us to directly tailor the safest and most efficacious cell delivery paradigm and dose to the DMD boys, which are obviously very vulnerable.
And so for me, that tells me that the time on station for the interventional cardiologist is much faster, much quicker versus, say, some of the other trials that are using localized delivery with NOGA, where you're really going to be on site for 2, 3, 5, 10 times longer. Is that fair?
So obviously not an interventional cardiologist myself, but what I hear from my colleagues and the thought leaders that we work with is that using this very standard approach, which is done millions of times per day across the United States and many more than that across the world, same way you get an angiogram or a stent put in place, is something that they do as easily as breathing. So they like this, and it's very adoptable. Our product is a frozen product thawed on the bench and pulled up in syringe and delivered. It's about a 20-minute procedure that ultimately at commercial stage will be outpatient.
And last question, as I compare and contrast All-Star and Dynamic, is there a difference in terms of the mechanism of action or modality? Because when I think of the post-MI environment, I think of homing signals and SDF1 and ischemic gradients, which I don't see as prevalent when I get into CHF. Does that matter?
Yeah, so, advanced heart failure is really a disease of fibrosis, right? You have sequential death of tissue, whether it be from a viral insult, some kind of pathogenesis of an idiopathic process, or in fact from an ischemic insult, and usually by the time you have heart failure, it's multiple ischemic insults. And so you're really treating the same thing. You're treating that stiff, boggy, expanded heart that has a lot of scar, and we do believe the mechanism of action will be identical.
How important were these factors in your discussions with J&J?
So, I can't speak for J&J. What I can say is they said that they felt that the fact that we had an allogeneic intracoronary delivery system, the cells came from the heart, we're going to treat the heart, that sort of made sense to them. And then I think they also liked the fact that we were manufacturing our own cells, we knew them very well. And of course, I can't underemphasize the thought leader bench that we have behind us. We have some of the best cardiologists in the world that are fueling these programs forward, and I think that was also very attractive to them.
Okay, you have permission to go to the next slide.
I have no more slides, and I think we're...
Okay.
So we can keep talking.
So let's talk a little bit about what are the next catalysts in the world of data that I should be focusing on as an investor around the company, and let's start there.
So we have a lot of news coming out, and as I said, we have many programs that are in place. So look for completion of enrollment of Dynamic in the first half of 2015. The Duchenne Muscular Dystrophy program, as I mentioned, will be putting in an IND very shortly, and all goes well. And with regulatory bodies, I think we all agree there's a little bit of luck and fairy dust that's involved. But we expect to be starting that trial also in the first half of 2015, at least announcing it. And we have our Cenderitide program, which although is not a regenerative medicine product, as I said, that program's wrapping up as well. So that will also be some very important news. And the strategy behind the development of that molecule and a conjunction program with our cells is something that will be very exciting in Capricor's very near future.
One of the emerging themes that I'm hearing already is a discussion about fast-track status in Europe and in Japan. So is that something that you're starting to look towards to see how you kind of move this beyond the borders of the U.S.?
Yeah, so one of the important steps we've taken recently is to hire a VP of Regulatory Affairs, somebody who has got a very long experience, 30 years in the industry, including some pretty large pharma experiences that is helping to govern our regulatory strategy and certainly fast track and breakthrough are all the types of designations that we're thinking about for our products.
And when I look at the utility of these cells, in the heart, are there applications beyond the heart that I should start thinking about in other areas of the vasculature?
We definitely think these cells have a very unique healing potential, and they have a unique expression pattern of extracellular markers, like I said, and activities that certainly would lend itself to other areas of the body.
So you have a lot of goals. I see 3 trials in front of me. You also have a small molecule program. You have $25 million in the bank. Strategically, when you think about moving beyond these trials, how active are you in the partnering landscape? How bound up are you with J&J, and for what indications, and what's also available outside of the relationship with J&J?
So J&J has an option to license the cells for adult heart failure and other cardiology indications. That option comes due at the end of the All-Star trial, actually the 6-month data, which would be an interim data point 60 days after. We have also a manufacturing relationship with them where we're working on some product development opportunities. Certainly anything outside that space is open for partnering, and we're excited at the opportunity to work with other people in the space.
So remind me, have you ever stated publicly what the terms are of J&J's opt-in? So J&J bought the option to license, but is that a negotiation based on data? Yeah.
So they paid us $12.5 million up front. If they choose to license it in at the end of All-Stars we've talked about, then it starts a stream of bio-dollars and double-digit royalties and sales milestones, and they take over the development costs of the late-stage clinical trials, which in adult heart failure, I think many of you know, are largely expensive and time-consuming. But in terms of what was the other part of your question?
Well, really, just you're in a position to negotiate with J&J, and you have deal terms set up. They haven't been announced, but
Right.
There's a significant deal to be had based on data. Just remind me, what's the market cap of Capricor today?
The market cap of Capricor today is around $140 million.
So $140 million. You're in 2 phase 2 trials right now in heart failure, which is clearly a $1 billion market. J&J's got rights of refusal to move forward, and you're funded through proof of concept on these trials.
That's
okay,
correct.
Terrific. Linda, thanks for joining us this morning.
Jason.
Thank you so much.
Thank you, Jason. It was wonderful.